Hello and welcome everyone to the corporate satellite of varvis, entitled Read Long and Prosper. And the next 1 hour and 50 minutes you will learn about efficient long-read data analysis and how it's done with the varvis software. We have 3 great speakers with us here today, who I will introduce a little later. Before we jump in, let me briefly introduce to you who we are. Team varvis, we are headquartered in Rostock in Germany, right at the Baltic Sea in the very north. And the company behind the varvis software is called Limbus Medical Technologies. We are a software development company and a medical device manufacturer. This is a picture of the lovely team. The varvis software is a cloud-based clinical decision support system and certified as a Class C medical device under IVDR. Let's remind ourselves why this is important. The IVDR regulates clinical diagnostics throughout the EU. Can you hear me properly? Am I too close to the microphone? Okay. Okay, I think now it's better. So, start again. The IVDR regulates clinical diagnostics throughout the European Union. And it's not only about the certification, but also about the classification. So the IVDR classifies medical devices according to the risk they pose to patients, starting from a low-risk medical device, Class A, up to Class D, which is the highest risk. By definition, every medical device, or also software and genetic tests that are intended to be used in genetic testing, prenatal testing, and cancer diagnostics are by definition a Class C. And this means that in order to obtain the certification, an conformity assessment by an external notified body is necessary. So this means that this notified body will test that the medical device is developed and operated according to the relevant standards. And of course, you only see some examples here. The list is quite long, and we're very happy that the varvis software was one of the first software to be certified as a Class C medical device under IVDR. And the certificate covers the whole NGS data analysis, so secondary analysis included, tertiary analysis included. It's also true for germline and somatic analysis. We are also happy and proud that we are currently leading the market in Germany. You see some examples of laboratory service providers and university clinics that use the varvis software in routine diagnostics. And this is just an overview what you can do with the varvis software. So it's a complete software solution for all clinical NGS applications. In the orange banner, you see the use cases. So you can analyze smaller NGS panels, exomes, genomes, also carrier screening, cancer diagnostics. And the newest addition to the portfolio of analysis is the long-read data analysis in the center. And for all these use cases, we offer tools to facilitate and accelerate variant interpretation. This is for SNVs, indels, and CNVs. For long read, also for structural variants and tandem repeat expansions. Where genomics data aggregation is included, classification alerts. Classification alerts means that for variants that are reclassified in ClinVar, you will receive a notification badge in varvis so that you can revisit these affected cases again. Raw data processing is automated and also validated, meaning that it's of proven analytical performance. And last but not least, we have expert support. So we have a very dedicated support team ready to answer all kinds of questions, ranging from IT to bioinformatics up to feedback on challenging cases and extraordinary variants. So all this is covered by the IVDR certificate so that you can navigate the NGS universe with confidence. And that's it for a short introduction. Let's jump in to what the session is actually about. And we have 3 speakers, 3 talks that will talk to you the next hour. We will start with Dr. Hauke Baumann with his talk, One Assay to Rule Them All. And they will learn how he and the team at Amitis Genetics how they replaced Sanger and long-range PCR assays with a long-read targeted panel. Second speaker is Dr. Preeta Balakrishnan, and she will talk about T2T versus HT38 and how a journey to the new standard could look like. And last but not least, Dr. Ben Liesfeld with his talk Performance Matters, especially with respect to long-read data analysis in a medical device software. We do have questions, time enough for questions and answers, maybe also in between, but definitely at the end. And so I'm happy to hand over to Dr. Hauke Baumann. I'd shortly like to introduce you first. So he studied biology in Würzburg and Kiel, and after obtaining his PhD in neurogenetics in Lübeck in 2020, he decided to turn his back on scientific research, but to dedicate his passion for genetics to patient care. So then he started working as a scientist at Amedis Genetics, involved in routine diagnostics. And his current focus is on establishing a long-read sequencing enrichment approach in a routine diagnostic setting. Welcome, Hauke Baumann.